The ingredients that make it differential will be the sugar mannitol, whether it ferments it or not. Okay, so that's what he wants for the answers for that.
Okay, thank you.
Next, it says describe the different growth patterns that Staph epi will show versus Staph aureus. So, what color was Staph epi going to be on the mannitol salt?
Green?
Pink.
Oh,
pink.
pink.
You know, and then Staph aureus that does ferment the sugar and makes an acid. Staph aureus will turn
Yellow.
Good. So we're going to put down here. What do you want to say? Pink and yellow for that. Next, we're going to talk. Are you recording?
Yes.
Okay. Next, we're going to talk about how bacteria grow on blood agar plates. So we have blood agar plates that I purchased from a company. We don't use human blood. We have to use sheep blood. And basically, the breakdown of blood cells is called hemolysis. And there are 3 different types of hemolysis. Hey, how are you? There are 3 different types of hemolysis that you have to differentiate between. So you have to differentiate between alpha, beta, and gamma. Okay? So if it is beta hemolysis, it means that there is a complete breakdown of the blood cells, and you are going to see very clear areas surrounding the bacterial cells, the bacterial colonies. So if it's a complete breakdown, you're going to get a clear area around it. If it is alpha hemolysis, it is only going to be a partial breakdown. So you're not going to be able to see through here. You can see through it. This is a partial breakdown of blood cells, and it kind of has a green hue or a green sheen because it's not a complete breakdown. In gamma, there is no breakdown of the blood cells. There's no hemolysis. It just looks like tiny little regular dots of bacteria growing on the surface, just tiny little colonies growing on there. So here it says, give some examples of an alpha and beta hemolytic example. So for alpha, it's going to be strep pneumoniae that causes pneumonia. And for beta, it is going to be strep pyogenes that causes strep throat. So you need to memorize that an alpha example, strep pneumoniae, and a beta example would be strep pyogenes. Next, what is blood agar? Explain what hemolysis is. Blood agar is just a nutrient media that has blood in it. Hemolysis is going to be the breakdown of blood cells, whether it is complete, partial, or no breakdown of the blood cells. And then it says, be able to recognize the different types of hemolysis as they are seen on the blood agar plate.
Yeah.
We have very good examples that we got. So you'll see one of these. It's going to be fine. He'll tape the edges, and you can hold it up to the light, and I think you're going to see the results better than if you just set it at the table. But he'll have a little card there that says you may pick up this plate to observe. Okay?
Yes.